All right. Welcome everyone to our TNT seminar today. Today we have Dr. Tim Gardner from the University of Oregon speaking, who is chair in neuroengineering and a founding member of Neuralink. We're very excited to hear his talk. And before we hear from him, I just want to give a couple, intro notes. So for those who are new today, TNT is the Translational Neuroengineering Technology Network at Hopkins, and so it's an interdisciplinary group for anyone at Hopkins or beyond who is interested in neuroengineering, especially translational aspects of it. And we put on events like this, academic ones, seminars, journal clubs, and we also put on social events like happy hours and professional development events. So one event I'd like to highlight coming up very soon, next Friday, February 3rd, is a professional development all-day workshop that we're putting on. Mostly our main sponsor is BlackRock Neurotech. And so it's going to be a really exciting day. We'll have a keynote speech from someone at BlackRock. We'll have a panel with members of industry from BlackRock, Ripple Neuro, Abbott, and the Applied Physics Lab. And then after that, we'll have lunch for everyone who's there. And in the afternoon, we'll have some tables set up to meet with the different companies and also to hear from the Professional Development and Career Office and the Johns Hopkins Tech Ventures Office to learn about resume tips, interviewing, entrepreneurial, incubators and support here at Hopkins. So it should be a really fun event. We hope you guys can join. And I'll post a link at some point for the RSVP since we need to get the numbers. So that's kind of our really, really big event coming up. And we hope to see you guys all there. Okay, so a couple notes for this event now. You're welcome to jump in at any time as you have questions during the talk, so please do. And Dr. Gardner said he's very happy to answer questions as they come up and clarify. So feel free to do that. Secondly, there will be a poll near the end of the talk, so please fill that out. It helps us to know who's coming to their events and how we can better serve you guys. And so with that, that's all the logistics out of the way, and I'll let Dr. Gardner take over from here. Thank you again for joining us.
All right. Well, thank you very much for the invitation, Mark. It's really a pleasure to be here, and I'd like to know more about what everyone is doing here. So it sounds like a really interesting program. But I'll just dive in now with introduction to the folks involved in this project. So I'm going to be talking about some new work that is mostly developed by team members shown here at the University of Oregon. And then I'll mention some recent work from Tim Achi, who we worked together at Boston University prior to University of Oregon. So just by way of introduction, the kind of main message for the talk today, I'm going to talk about a new thin film electrode array fabrication method that the lab's been working on in Oregon for the last couple years now. And it's something that is kind of pre-publication. We have a preprint out on the subject, but still early days on this project. And I think by way of introducing that, I thought I'd tell a little bit more of a story of how we got to working with this and some of the real interests of the lab going beyond this particular device. So my lab historically has studied sensory motor learning in songbirds. And this is a well-established model that some of you may know about. There's a really well-defined circuit that involves both cortical-like areas and basal ganglia areas of the brain, and interactions between the two is really a strong interest of mine, and that relationship between cortical and basal ganglia circuits are particularly well-defined in the song system. And in recent years, we've been shifting to focus on a new species. So this is the canary. It's a bird that has incredibly rich songs, 50 or 60 syllables, 50 or 60 seconds long, and really complex syntax. Beautiful singer, and it's showing some really intriguing new principles about the relationship between basal ganglia and cortex in the production of song in real time, something very different than what we've expected from zebra finches. I'm not going to talk about any of that today and really focus more on a kind of a history of the challenges we faced and some of the things we developed in the process of trying to record better signals from small brains. And we believe these are challenges that ultimately will translate into interesting human products where miniaturization is really important. So, one of the things that we've worked on over the years is developing the wireless version of a miniature fluorescence microscope. So these are bird-sized fluorescent microscopes similar to those produced by the company Inscopics. We made a version in the lab that is wireless. So we've since recorded in birds and bats with these free-flying bats in collaboration with groups at Berkeley. And the data that generates is pretty fun. So I just thought introduce a little data. I don't know if you guys were able to hear that or see the video, but flashing cells, very stereotyped activity as a bird sings. And this is a freely moving bird that's using actually just off-the-shelf wireless transmitters for first-person view remote control airplanes. Those were the sensors and the wireless devices that we built this out of. So the lab has faced this challenge of how to miniaturize, and the Inscopics microscope was a little bit too large for us to use, and we really needed more mobility, and found this really low-tech, low-cost solution in 3D printing and then first-person view wireless transmitters for remote control airplanes that would give us this free-moving, free-flying recording and singing birds. And so to make a long story short, we found this really interesting pattern when we would use these to describe the activity of the cells that participate in song. And what we see here is that on different days, even though the song is not changing, we found different neurons encoding the song or participating in the production of that song. So a cell, for example, that's red in this image, the GCamp calcium activity is visible really only on the first day of our recording. And on day 2 and day 3, it's gone. So the only things that persist across multiple days are these white cells. But the blues, greens, yellows all indicate that there was some degree of turnover in the participation of these cells in the production of song. This really fascinated us because the song itself is stable at this point. The zebrafinch we recorded from is just producing a rock solid song every day. It's his identity as he sings, and there's no evidence that he's in the process of changing that song. And even so, in this incredibly stereotyped activity, we see pretty massive turnover of the population of neurons that produce the song. Even more interesting, almost all of that turnover took place over intervals of sleep. So it really suggested that the brain undergoes significant reorganization even in the production of a stable memory pattern from one day to the next. Now we wanted to investigate that further, but looking now at cell types that we couldn't image with the calcium indicators. And this is just a panel that shows from the work of Michael Fee, his lab at MIT, recorded from projection neurons in the song system. So these are these sparse firing cells, and then inhibitory interneurons. And you can see that these two classes of projection neurons are very sparse. They're really well adapted to calcium imaging with relatively slow indicators like GCamp. But the inhibitory neurons have the denser firing rate that we couldn't resolve with the slow timescale of the calcium indicators. And so that turned us back to electrophysiology. And we can't just use the Utah array at this point because it's quite large relative to the scale of the bird, but that's really the kind of format that would be ideal for recording from this thin nucleus that we'd like to sample from. So in lieu of being able to use commercial devices, this is now close to 10 years ago, we built a carbon fiber electrode array. This was inspired by the work of TK Kozai, who had made single channel carbon fibers and showed successful recordings in mouse brains. And we just generalized this to a couple changes. So creating sharp tips on these carbon fibers and then bundling them together into these little tight bundles in a little 3D printed electrode. And to our surprise, when we would insert these in brain, bundles of fibers like this, we would see a gradual spreading or opening of these fibers upon insertion. So those bundles wouldn't stay stuck together, but they'd actually open up and be deflected by the microstructure of tissue. And you can see here examples of sub 10 micron fibers inserted in a bird's brain with new end stain. This is at a depth of 3 millimeters below the surface. And then here's a really large bundle of fibers that we inserted, a few hundred of them, also at a depth of 3 millimeters below the surface. And so there's this really interesting surprise that these very, very flexible small fibers can be held together to be stiff enough during implant, but then they spread apart during insertion. And what that allowed us to do is basically get the very first chronic signals from inhibitory neurons, so stable recordings from these dense firing cells. And these are very small neurons in the song system, typically ranging from 8 to 12 microns in diameter, challenging to record single units. So we were really excited by the yield from these carbon fiber electrodes and stability in this outlier case of 124 days. And that really kind of closed this interesting story on the, or I don't know, closed it, but put a counterpoint to the excitatory neurons that we had recorded previously. So in contrast to those excitatory cells that drift in and out of participation over the course of a day, these inhibitory neurons we found to be really rock solid stable for as long as we could record them in terms of the firing patterns. Similarly, we were able with these carbon fiber electrodes to record excitatory neurons. And in that case, we could see instances of drift where an individual cell might begin a single day with one burst of action potentials per song in a stereotype position, as shown here. And then towards the end of that single day recording, a new time would appear, and that cell would now fire twice per song. And so consistent with the kind of drift that we've had described previously in the excitatory neurons. I'd say that story about the stability of memory and relationship between different cell types in preservation of memory is still an open story. There's a lot that we'd like to address further. But on focusing on the technology for today's talk, the carbon fiber electrodes were something that we found useful. I would say relatively low yield in that we get 1 or 2 stable neurons per bird that we would implant, and we would really like to have had many more of those. They were just extremely difficult to build and build consistently. These were all hand-built devices involving a pretty boutique process. And so that led us to collaborate with Stuart Kogan's group at UT Dallas, where, and with the support of the NIH, we developed a concept which was silicon carbide needles that had this very similar modulus and geometry to the carbon fiber electrodes that we had been implanting, but these could now be microfabricated in a clean room using more of a high throughput scalable process. And that work is still in development. Stuart's lab continues with that, and Felix Deku, shown here, was the PhD student that worked on that project primarily. He is now at the University of Oregon, my next door neighbor. We were able to recruit him just this last year, and we're really excited to continue pushing forward these devices because we think that they have both applications in small animal models, similar to what we had used them for, but also really interesting clinical applications because of the material that these are made out of. They're silicon carbide, which is one of the very few materials that the body doesn't appear to degrade upon implant. So the longevity of these with chronic implants and accelerated aging looks really good. And we think that they could be particularly useful for stimulation in cases where you need really small scale stimulation for therapeutic purposes with high channel counts. So if that's of interest to any folks here, I'd be really happy to talk further. We're in the process of kind of restarting this research program on the silicon carbide needles at the University of Oregon. And some of the approach that we're going to take to that is informed by work that we did at Neuralink, and I'll switch to just talking a little bit about that. So around, I guess now about 7 years ago or 6.5 years ago, I left Boston University to work as a co-founder with a great group of people at Neuralink. And about a year or two after starting there, Felix also joined and ended up head of microfabrication at Neuralink. So we took some of the thin film devices and skills that he had developed with Stuart Kogan at UT Dallas into the Neuralink stack. So just to review, for those of you who aren't familiar with the Neuralink project, there's probably no one who hasn't heard this in some form or another. The company's evolved quickly, but at the time when I left, now about 3.5 years ago, we had built this surgical robot, which was designed to move a needle, a very precise, small needle that here is 25 microns in diameter, with a little notch illustrated here. That needle was designed to hook individual electrodes that you could see fabricated here. And Felix made these with Parylene backer. And so you essentially would peel off the thin film polyamide electrodes using that little loop at the end. And then the surgical robot would image the vasculature using OCT and other reflected light modalities to avoid blood vessels and gradually sew electrodes into the surface. So this figure here kind of illustrates that process. After peeling off an electrode, that needle pushes forward through a cannula and then retracts, leaving one of these extremely thin, flexible electrodes sewn into the surface. This here is just a kind of a close-up of one of the earlier versions of that inserter head with the sharp needle, and then this pincher, which was used to peel off electrodes from the Parylene backer. Currently, the design doesn't require that, so it's just a needle. And this worked really quite well in rats. And at the time that that I left the company, I would say that the emphasis had shifted a bit from trying to scale up to as many channels as we could possibly do to more of the human product and the wireless form factor. But in the early days at Neuralink, we were pretty focused on trying to push channel count as far as we could go for small animal models. And that's kind of illustrated here through a series of evolution of devices, beginning with early test cartridges through to this device, which I think was our probably highest channel count implant in a rat, and that was 3,072 channels in a rat, just connected with a USB-C cable. Any questions at this point? I've been going kind of racing through lots of stuff, so yeah. All right. Well, that was Neuralink. That was a couple-year experience for me, and I would really love to see these devices or some version of this made accessible to the research community. I think particularly for our work in mice and songbirds and small animals, as well as large animal models. I think there's a really nice flexibility here to essentially insert many electrodes over a relatively wide region of cortex, and to do so in a way that's minimally invasive. So designed to miss those blood vessels on the surface of the brain. And this works particularly well in cortex, where once you get beyond those first surface blood vessels, the most of the remaining large vessels are perpendicular to the surface. So if you can orient and insert perpendicular, then your chance of a really significant bleed is pretty low. So I think that there's a real opportunity for the research community to kind of restart this process in a form in academic setting. I think the the thin film fabrication is something that Felix Deku and I are working on at University of Oregon, and in collaboration with Texas as well. And the inserter head, the needle that can sew in these devices, I think is really quite straightforward at this point for us to fabricate that. Hardest part is the electronics, right? The Neuralink's custom ASICs are not things that we'll have access to in the academic community, but I think there's things happening, and certainly beginning with Intel chips and things like that is something that I think is really worth doing at this stage. So this is all work we'd like to be extending at the Knight campus. This is my current workplace. This is the University of Oregon, and a new bioengineering department at the University of Oregon. It's the first engineering department at Oregon. For historical reasons, engineering and medical school and basic sciences were split onto 3 campuses in 3 different cities, about 3 hours apart. And so it's making interdisciplinary work quite a challenge. So for the University of Oregon, this is really a big deal to have its first engineering department. And so we're pursuing neural engineering as a part of that. And then I would say the major center of mass in terms of research interests here is regenerative medicine. So there's a lot of folks that are working on regenerative medicines, tissue scaffolds, particularly for musculoskeletal applications. So the neuroscience group is a small group here, but we hope to grow that in the future. All right, so that was kind of a little bit of a history of where we were for the last few years and things that we were working on. The consistent motivation has really been to try to improve the quality of signals, the stability of signals in small animal models, and to do so by miniaturization and really making more and more usable, more scalable approaches to electrode insertion or microscopy. So for the next section, and
I do have a question.
Yeah, please.
So I guess in some experiments I've done with rats, when we're trying to pick electrodes, I think a big limitation has to do with the price piling up because of I guess, like the electrodes not being multi-use. So I guess with some of these designs that you're talking about here, are they intended to be chronic, reusable, or they're just like acute one-time experiments? How do you balance those different factors?
Yep. Yeah. So I think everything that I've talked about so far on the electrode side is, mostly single use has been the way we've used these. And I think that this is a real issue. I anticipate for the current electrode projects that we're going to move through the NIH U01 type distribution channels, which would fund networks of collaborating labs, people that are interested in these. But there's a real valley of death challenge for getting this kind of neurotechnology into the market at affordable prices. And so I think, I don't have a good solution for that, other than really grateful to the Brain Initiative and U01 type initiatives that can get things out into testing, but it's still not widespread markets. And my hope is that we can get better data and just require fewer animals. So that, there's, I think that's part of the solution too, is that because some of these things are intrinsically, somewhat time consuming or labor intensive to build, we really need the good quality signals and as much as data as we can possibly get, and then it's worth it. So it's kind of a hand wavy answer, but get away from the regime where our earlier work with the songbirds were getting 1 neuron per bird, and really try to, can we get 50 from one recording, and then let the paper involve 3 animals instead of 30 or 50 or 100.
Right. Okay. Cool. Thank you.
I don't have a good answer for you.
Yeah, it seems like a challenging problem, and I mean, for us, if the animal dies mid-experiment, then it's like, or you just get you don't get the greatest data that day, then it's an expense and time and all that kind of unfortunately goes to waste. And how do you make sure that your chance of success is high every single time? It's not in part of my main work. So it's something I'm like collaborating with others, and I feel like that's been a stressor for sure.
Yeah, I agree. I don't have a good solution for that. I would say that what continues to motivate me a lot is just a dissatisfaction with the quality and number of cells we've been able to record in our behaving animal studies. That it's just been um, it's still not where we'd like it to be. I'm
Mhm.
excited to be using the new neuropixel arrays as those are reaching the. I think NeuroPixel 2.0 is going to be small enough that we can routinely use those in songbirds, but there is a real cost item there, and we know that those signals don't persist more than about 24 hours in songbirds. That they're hopping around, moving their heads a lot, and makes it pretty challenging to hold high quality signals stably. So we really want that, and so I'm with you. But I don't have a silver bullet for it yet. Um, so the next section, we're going to talk a little bit about micron resolution 3D printing, and this is a new method that we're using for fabrication of electrode arrays. Um, the the process is probably familiar to many of you, but the principle is just quite straightforward, similar to two-photon microscopy. And so in two-photon microscopy, you use two infrared photons to excite your fluorophore, and the probability of absorbing two infrared photons is extremely low, except right at the focal point, right? So you've got this strong nonlinearity that makes it possible to excite fluorescence at a single point in a fluorophore rather than the broad spread of fluorescence excitation that you would normally get with single photon lasers. And the um, principle is just that these pulsed lasers, bunch up all of those photons together into extremely brief pulses. I like to remind people that a femtosecond is to a second as a second is to the age of the universe. So it's a really short pulse of light, and some pretty cool stuff happens on those short time scales. The same principle applies to polymerization. So you can scan your laser through a liquid photoresist, and with those femtosecond lasers and pretty standard ultraviolet photo initiators, you can polymerize structures with extremely high resolution in 3D. And there are companies that produce printers. One of the first out there was the Nanoscribe. Many of you have probably heard of or used before. The approach that we took to fabricating in this two-photon space was to modify a two-photon microscope with a resonance scanning mirror so that we could do printing on a faster time scale. And it's essentially just a two-photon microscope running ScanImage, which is that open source software that some of you may have used for two-photon imaging. And we made a plugin that goes into ScanImage that basically converts any two-photon microscope into a 3D printer. And what's pretty cool about that is the standard two-photon microscope these days include resonance scanners, and those can be typically 8 kilohertz, sometimes up to 20 kilohertz for some of the fastest ones these days. And so instead of steering the beam as Nanoscribe does, where you have just two galvo mirrors that point the light, relatively slow moving mirrors, we do this fast raster scan where the laser just scans out a stereotype pattern, raster scan like the old fashioned televisions, and then it turns on at specific pixels. And this turns out to be somewhere between 20 to 100 times faster than the Nanoscribe device that was pictured here. And that really was a game changer for us to be able to efficiently use this to build devices for the lab. So there's a little plug-in 3D printer. You design your parts in STL files, and then you can print them in with the two-photon. This was an early version of a nerve interface that included carbon nanotube threads that were manually placed on the device, and just the speed of printing really allowed us to efficiently explore many dozens of designs for some of these projects. The other thing worth mentioning is that same software and package can do efficient laser ablation. So if you think about laser cutting, typically it's a really complex process. You're generating a lot of heat. But if you go in with femtosecond pulses, particularly high amplified high energy pulses, you can just tear apart the atoms in a way that leaves no heat affected zone. So it just vaporizes the material, and you can get really, really smooth laser milled surfaces. And this is just an example of some very precise pits that we cut on silicon wafers using a Monaco laser. This is a amplified ultrafast laser, and it's the way that I built the needles for Neuralink, where we use a raster scanned laser mill that allows us to achieve extremely high resolution in the needle shape as shown here. All right, so for the Songbird work, the first device that we sought to build with this approach was a nerve stimulator. And so the nerve that drives vocalizations in songbirds is about 150 microns in diameter, really small and fragile. And this, at the time, was the smallest device that we could buy commercially to interface with that small nerve. This is a Cortec 150 micron diameter, or rated for 100 micron diameter nerves. And you can see it's, maybe that interior diameter is 100 microns, but this is the scale of the device that we're able to build with the 2-photon lithography, and just much more manageable on a very small nerve in a small animal than this larger bulky structure. That later was refined to include all microfabricated electrode traces. So instead of using that carbon nanotube thread from Rice University, this was Matteo Pasquale's magnificent, amazing material, we went to something that was fully microfabricated, so just a polyamide electrode with gold pads, and then we could 3D print on two sides of that thin film device, and that create a mechanical clip that would hold a thin film electrode onto a nerve. And that's the finished product in size, and then again a kind of comparison to the commercial state of the art for nerves of that size. And so we found with the really tiny nerve devices that really, to our surprise, we got quite large signals. So signal to noise ratio higher than anticipated for recordings outside the nerve. So signals up to close to a millivolt recording from the nerve. And one of the things that allowed us to use this effectively was having those 6 contacts with microfabricated structures. We could do really effective common mode subtraction of those signals. So take the average of all 6 electrodes and subtract it from each individual electrode in order to erase movement artifact as much as possible in recording from these small nerves. And this is an image of songbird vocalization, just 3 syllables, and then the raster plot of activity recorded from the surface of that nerve across 30 days. And so we're pretty excited that this is like a first proof of principle that you really can interface with nerves of this size. And we think that there could be some really interesting clinical applications in small animal, preclinical models and in mice. And there's groups like GlaxoSmithKline and Galvani that have actually supported this work. I should have mentioned them in the beginning, to develop nerves for preclinical testing in some of their projects. And just to name a few of the nerves of interest, carotid sinus nerve in a mouse is involved in regulating cardiovascular system, and a really important clinical target for diabetes and other forms of cardiovascular disease. It's only 50 microns in diameter, right? So it's really hard to get a sense for how the brain and the body communicate along those nerves. Other nerves, splenic nerve in rodents that is now a really interesting target for rheumatoid arthritis. And you can just go down the list of many nerves of interest for bioelectric medicine in small animal models are extremely difficult to record from. And from my perspective, it's really a wide open field, right? That how signaling takes place on some of these nerves in the periphery in freely moving animals that are really recovered from anesthesia and stabilized post-surgically. As far as I can tell from most of these medically relevant nerves, there's very little information about the activity in those nerves in a natural setting. So stimulation, that was our initial goal. We developed a paradigm where we could blow air through the syrinx of the bird and then stimulate on that nerve to induce different vocalizations. And here you can see a series of 3 or 4 vocalizations that were produced by taking essentially the same pattern on that little nerve stimulator and then just inverting it. So pattern A, or inverting it spatially, pattern B, again, inverting it spatially, and you can see, distinct vocalizations. So indication that there's some power to control a nerve with variable stimulation patterns. One electrode is defined as the source and another the sink. So it's a biphasic, bipolar, or multipolar stimulation, and you can just do anodal first stimulation on one contact and cathodal first on another, and that defines a little path for current steering between those electrode contacts and gives you pretty good spatial control, apparently, over the activity within that nerve, at least to the degree that we need to drive different vocalizations in the songbird. And that's just quantified here, where each of these colors is a different spatial pattern of stimulation on that nerve, and then this is a T-SNE plot of reduced dimensionality, indication of each cluster showing a different acoustic structure, different vocalization is quantified acoustically. All right, so getting to, if there are no questions on that little bit, this was our first 3D printed electrode array. It's work that we're continuing and would be happy to collaborate with folks in this space if folks have need for small nerve devices. The next goal was really to try to build something that would compete with the Utah array directly in form factor for cortical applications, possibly also for nerve applications. And we know, things on the scale of that Utah array have a pretty big glial response. There's a lot of chronic challenges in using that over the long time scale. We were excited by work that was published by Carnegie Mellon. This is the Carnegie Mellon array. It's a 3D printed electrode array that uses an aerosol droplet of metal that can kind of build 3D arrays with slightly different, very different process than the Utah array. And we asked if we could do something similar with 2-photon lithography. And the reason that's highly desirable is that 2-photon lithography resolution is just another order of magnitude better than what you could do with aerosol jets. So if you really want to make extremely fine features, lithography, 2-photon is useful. So we spent a couple years on this. My lab has more microfab people in it than it has birdsong people in it right now. This ended up becoming quite a challenge for us, but the biggest one is that the standard microfabrication processes that involve photoresist are developed with a developer that will also dissolve the 3D prints. So if basic idea is start with a thin film electrode, at some stage in that process, incorporate 3D printing, and then take it back to the clean room, finish the electrode in the clean room. That historically has been a real challenge because these 3D prints are dissolved by the developers that are used in photolithography. So for us, we got this working just by using metal masks. Initially, this was a chromium mask for an important step in the process. And to make a long story short, we were able to fabricate these electrodes in standard standard cleanroom lithography steps for polyamide, and then 3D printing, and then metallization of everything after that 3D print process, followed by further liftoff steps that would remove metallization from areas other than the desired targets. And a final step in this, yeah, go ahead.
Yeah, so back at the picture of the electrode array,
yeah.
it looks like the tip kind of has this flat surface. Is that a design contact thing, or is that a artifact that you don't know what to do with it?
Yep, that was designed. So the last step in the array fabrication process is to coat everything in parylene for an insulator, and then we use the femtosecond laser ablation to reopen those tips to create recording surfaces. You can see here on an XPS, the platinum signature is in red, so there's platinum metal that's exposed after that laser ablation. And we just thought that adding that little bit of microstructure on the tip of the electrode would increase the surface area for the metal and lower our impedance. So we haven't tested this systematically, but our concern was that if we just had a, essentially a flat surface when we did that laser cutoff, it would be too easy to completely remove all the metal from the top of the surface to create that recording tip. So we added this little, because it's 2-photon lithography, you can get really, fancy with any structure you want to add to an electrode. And that so far looks like it's been useful. So here you can see in another image, these were the first prototype electrodes we built with that parylene outer coating and then the exposed platinum after the laser ablation.
Does
So this, yeah, go
does
ahead.
that have any like effect on almost like recording direction, where you have to be like very perpendicular, or the surface that you're interacting with, and it loses kind of like recording on the
Yeah,
side of the tip?
could. I think it could make things a little bit more directional. It's not something that I guess the next slide, we'd really like to get away from these flat top surfaces. This is still something that we're trying to develop further, and I don't know for sure if we're going to need that. But the next slide really illustrates the current challenge that we're faced with, and we haven't worked this out completely yet. But one of the variations that we were able to print, these were electrode spikes that have a little protrusion, sharp protrusion at the tip that was designed to mimic the structure of a mosquito proboscis. And these arrays we found could actually be implanted in small animals, mouse and bird, going through the dura. So we didn't even have to dissect away the dura, but could just take that thin film array, lay it on the surface of the brain, and then just kind of push on the backside and get it through the dura. We haven't yet made functional tips with that particular sharp feature, so we'll need to open a contact point on the side while still preserving that sharp tip. And I'm optimistic that we'll be able to do that, but hasn't been completed yet. But just kind of initial insertions, this is kind of what that array looks like. Bed of nails, similar to the Utah array, but now on a really flexible substrate with protrusions that potentially are quite sharp and thin. And current work in the lab is seeking to replace that parylene insulation with silicon carbide. So that's the material that we had mentioned previously. It's a really good insulator under certain circumstances, under proper deposition parameters. And it's also quite stiff. And so what that's going to allow us to do is reduce the cross section of these needles even further. And so the student in the lab that's working with that has gotten down to about 5-micron needle sizes that are freestanding, up to a 350 micron tall. And I think that'll allow us to increase the density of the needles with before we hit that bed of nails effect where the brain just kind of dimples when you try to insert it. So we're pretty generally excited about this form factor. I think it's something that I would love to be in communication with people interested in translational applications of this. We've thought particularly of targets like retina, possibly the sulci of the brain, if you want to try to insert something down into deeper sulcus in a primate or human application, and then potentially cortical arrays for whenever you need really high density interfaces in cortex, visual cortex, neuromodulation potentially, and nerve applications. So we think that these are going to be small enough, sharp enough that we can actually assemble them inside the nanoclips that the lab's built for nerve applications. And ideally that would give us now penetrating spikes that would go into these small nerves and further improve the signal quality that we're able to record. So just wrapping up, some of the kind of future concepts. The material we 3D print with is transparent. So it's essentially a photopolymerizable glass. It's a silicate with methacrylate groups called Ormacomp. And it's possible to actually do optical stimulation through the centers of these and in conjunction with the electrical stimulation. So here's a fully um, fully fabricated electrical array that is also transmitting light, and so mating some of the micro LED devices that are available now for virtual reality. On the backside of this, I think it should be feasible to do pattern stimulation. I don't think the powers are quite there yet to be able to use these micro LED displays, but I think that's potentially coming soon. These are just some other pictures of electrodes that we can build with that flexibility in geometry. So porous structures. This could be useful if you want something that integrates with tissue and has brain growing into it or some other bodily tissue growing into it. We're able to metallize the full interior of those. So the sputtered metal coats all surfaces within that. So you get, in principle, you can get much higher surface area for electrochemical charge injection than you would with just a flat, smooth electrode. That's one concept that we're interested in exploring. And then just kind of last slide in closing, we'd really be keen to collaborate with folks that would like to do tissue organoid type applications as well. So we see a lot of possibility for 3D printing structures with 2-photon. You can 3D print hydrogels for scaffolds. The lab's been working with. Here's some cells growing on a 3D printed array of pillars. And now if you can prefabricate electrode, small high density electrodes that go into these scaffolds, it might be interesting to be able to just seed neurons or other organoid models onto scaffolds that have integrated electrode arrays in them as part of the scaffold. So new concept and new directions, and would love to hear your thoughts. And if anyone wants to reach out, feel free to after the talk as well. And that's what I had for today.
Anyone have any questions that they want to ask before Tim goes?
Well, I'll give you my my email. It can be found pretty easily at University of Oregon. So it's timg@uoregon, and we're always eager to consider new applicants for PhD program or for postdoc. And if you've got a company idea, something you'd like to spin out that connects to this, really be happy to talk and hear about what you're interested in.
All right. Well, thank you again so much. It was very interesting, and I think we saw a lot of stuff probably that's new for us, new configurations. I feel like for a lot of our experiments, it'd be cool to get our hands on this in some way. So thank you for the time.
Yeah.
Thanks
for the invitation, Mark.
Yeah, our pleasure, and hopefully we get to keep in touch in the future.
Sounds good.
All right.
Well, have a good day. Yeah, we'll
Thank
talk to you later.
everyone.
Bye-bye.